But I think we made it through Friday the 13th, and let's hope that the next couple days are sunnier and without incident. So again, thank you, Larry. Thank you, Kathy. Thanks, Tom Miranda, and everyone for having me here. I'm going to share my screen and go ahead and get started. Oops, go back. Can you hear me well enough,
Yes.
everybody? Great. Okay. So this lecture is going to probably be a little different from what you're accustomed to. I will talk about some scientific aspects of my research on this genus of orchids, Porroglossum, but at the end, I'll also offer up some horticultural tips because I think everybody should give a try growing these really amazing little plants. And I also want to give credit to the man who I'm standing next to in this picture. This is one of my former PhD students. His name is James McDaniel. I was hoping he'd become an orchid biologist or a professor, but he decided to follow the money, and now he's working in human genomics out in Los Angeles. But most of the work that I'm going to be talking about today was part of his PhD dissertation. And I thought I might give you a little bit of background about myself. I know when we have orchid speakers come to our club, I like to know a little bit more about their background or and where their passion for orchids came from. So I've been an orchid nut since I was a child. I grew up in Detroit, Michigan, and even at the age of 9 years old, like in this picture, I was fascinated by orchids. And I remember on my birthday, instead of a catcher's mitt or a baseball bat or something like all the other little boys, I asked my parents for an orchid. And in those days, it was hard to track down orchids in Detroit, but they found a beautiful Paphiopedilum for me. You can see how happy I was. I'm sure I killed it shortly thereafter, but I've learned a lot since then. Um, from Michigan, I moved to Tennessee where I was educated, high school and college. I then spent a year at the Smithsonian Institution, working for the Natural History Museum in DC, and then went on to graduate school in North Carolina, did my work at Chapel Hill, and then ultimately ended up at the New York Botanical Garden for 10 years. And along the way, I spent 1 year at the Royal Botanic Gardens, Kew, doing research on orchids at Kew Gardens. But the Upper Midwest called me back home about 20 years ago. And so I have become a cheesehead, basically, now living in Madison, Wisconsin, where I'm as Kathy mentioned, not only the herbarium director and a professor, but I also oversee our conservation biology major on campus. We have about 200 students who are majoring in conservation biology of plants and animals. So conservation is a passion of mine. And this year, I'm also the president of the Botanical Society of America, a 3,000 member organization for professional botanists in the US and around the world. So I wear a lot of different hats, but it's the orchids that are my true passion. If you've been a member of the AOS for a dozen years or more, you might recall back in 2013 that my students and I presented a series of essays in Orchids magazine. Each month we featured a different group of orchids to profile their evolution. And our goal in doing that was to try to bridge the gap between the horticultural community and the orchid scientific community. I think we have so much to learn from each other, but we often don't go to the same conferences and we don't interact as much as we should. So I think we could do a better job of that. If you don't remember those essays, you might know about my book from 2011 called Vanilla Orchids, Natural History and Cultivation. My primary research focus is on the biology of vanilla. That's what I've been doing for the past 30 years. And in fact, I spoke to three different orchid societies on Oahu some years ago about my research on vanilla. And I was really happy that yesterday I got to tour the vanillary here in Kona. And it's really exciting to see that I think the first and only commercial vanilla plantation in the United States is right here on the Big Island and seems to be doing really well. So that was really exciting to see. I also wrote a book years ago called Orchid Biology: Reviews and Perspectives. That's very scientific. And later this year, I'll be publishing the ninth edition of The Biology of Plants. This is the world's best-selling botany textbook. It's been in existence since the 1960s, and I'm really honored to be carrying on the tradition into a ninth edition. So if you have kids or grandkids who might be in college and take a botany class, hopefully they'll use my textbook starting next year. So along the way, I have done a lot of research, published over 150 papers, scientific papers on a whole variety of different orchids. Some of these I know you recognize, like Dracula and maybe Liparis, vanilla, obviously. But I tend to like the weird orchids more than the common ones. I think they're all beautiful and fascinating in their own right. And the one with the square around it, Porroglossum, is what I'll be talking about today. I also want to mention that I'm not just a slicer and dicer who does research on orchids. I also grow orchids myself. One of my great passions is the Japanese Fukiran. These are all varieties of the same species, Neophonetia falcata or Vanda falcata. And I've got about 200 or 300 of them. And I was featured on the cover of a Japanese horticultural magazine because of my interest in growing the Japanese and Korean Fukiran. I was an exchange student to Japan in high school, so I've always had a passion and a fascination with Japanese culture, and bringing this into the orchid world is a perfect fit for me. I also am very actively involved in my local orchid club in Madison, Wisconsin. We are really growing like gangbusters. We've got over 150 members now, and around 80 come to each monthly meeting. I'm the treasurer. I'm also the webmaster, and I'm also in charge of the designing our exhibits. And this was our show exhibit a couple months ago. And I was very happy and proud that we received a silver certificate from AOS judging 2 years in a row. We're still missing that gold. I can't seem to break the 90-point barrier, but we were up to 84 points this year. So I think next year is going to be our year for the gold. But I only bring this up because I know that like you, your orchid club brings you a lot of joy, and you get to meet people from all walks of life, and that we have this passion in common, which is orchids. So what I'm going to be talking about is what I call the power of movement in orchids and relating to floral snap traps in the genus Porroglossum. And the reason I called it the power of movement in orchids is because Charles Darwin actually wrote a book by that name, The Power of Movement in Plants. Now, I know we all think of Darwin and Galapagos finches and tortoises and animal evolution, but I would argue that Charles Darwin was a botanist more than anything else. Right after he published The Origin of Species in 1858, he followed it up with a book on orchids, on the various contrivances by which British and foreign orchids are fertilized by insects, and then followed that book up with a number of different botanical themes, including several books about movement. He seemed to be very interested in how plants could move. And the movement of organisms, whether they be animals or plant, is known as kinematics, right? Like kinesthetics or something. Kinematics is the study of movement. And there's millions of studies on animal movement and humans in particular, with athletics, for example, but very few about plant movement. A few that I am aware of are studies on carnivorous plants, like the sundew that has tentacles that can move and capture prey, or the Venus flytrap. There's a few papers on orchids. For example, you know that Catasetum can eject its pollinia with great force. And people are fascinated by the kinetics or the kinematics of that particular movement. But what I'm going to introduce you to is another little orchid that to me is equally remarkable. And I'm sure that if Darwin knew about this plant called Porroglossum, he would have written a whole book about it. So this is a little species of Porroglossum. This is Porroglossum rodrigui. It has quite attractive foliage. You see the leaves there. But we zoom in on the flower and it looks a little bit like a Masdevallia, right? The three sepals are fused together into a triangular shape and there's kind of a hood there. It's got this unusual lip, though. The labellum is sticking out, almost like it's sticking its tongue out at us. And here it is in the face view. And most of the time we see it photographed like this, but oop, it can close its mouth. It can pull its lip in or up, um, depending on pollination biology. So what we think is, so here's another photograph of a different species, Porroglossum nutibrata, that has its lip in the open position, waiting for an insect to come, and then in the closed position after an insect has triggered it. What we think is happening here is that fruit flies, small flies, are attracted to the plant, probably a fragrance of some sort. It may not even be detectable by the human nose. And as they crawl around on the labellum, on the lip snaps shut and captures one or more flies. And as they wiggle around trying to escape, they pollinate the flowers. So it's a trap, but it's not eating or digesting them the way a carnivorous plant would, right? It's all about pollination biology. And so there are some detailed studies of the hinge that allows this movement to take place. And I do want to mention that I know you're familiar with some species of bulbophyllums and some maxillarias, and there's a number of orchids that have a hinged lip, right? It can kind of flutter in the breeze. We're not talking about a passive hinge here. We're actually talking about a trigger that snaps shut and then needs time to reset, perhaps even 24 hours later to reset. I think the best way to visualize that is through videos. So I'm going to show you three different species. Here's Porroglossum muscosum, and we're going to watch a video for a minute. I'll take a little toothpick and pretend I'm a fly, and I touch the lip, touch the labellum, and within a couple of seconds, it slowly closes, right? Now it's fully closed. So that took maybe 3 seconds or so for that to happen. Here's a different species. This is Porroglossum rodrigui. This one's going to be much faster. So I'm going to touch the lip with a, oh, and there it goes. Like in a fraction of a second, it snaps shut, right? This isn't just a passive hinge. This is like a Venus flytrap closing. And then Porroglossum hertzii is going to take a little bit longer, but we'll do the same thing. Imagine we're an insect. We're walking around on the labellum. We've triggered it. You notice the labellum is kind of made a slow movement there for just a second. It's slowly moving. It doesn't want to scare the insect away. So it's kind of slowly moving, stopped for a little bit. The insect keeps working, and then it snaps shut. So we've seen three different examples of a kinematic pattern that these orchids use in their pollination biology. So what I'm going to show you in a few minutes are a series of data where we took high-speed video, we took a video camera into the greenhouse and we recorded thousands of videos to quantify or to estimate the amount of time needed for the floral trap to snap shut. So both of these species shown here took around, a minute and 50 seconds or so, almost slightly under 2 minutes, like 1 minute, 53 seconds there in the bottom. And so we'll come back to that in a minute, but just a little bit of background on Porroglossum. It's a member of the subtribe Pleurothalidae. We call those pleurothallid orchids, right? And you probably know that this is the most diverse. It's the largest subtribe of the orchid family. There are at least 4,000 species of pleurothallid orchids in 29 different genera. That's 20% of all orchid diversity. These are only found in the New World tropics and mostly at high elevations, so especially in the Andes mountains in the cloud forest habitats. They're probably less frequently cultivated than most orchids around the world, just because they do require that cool temperature and moisture, and a lot of us tend to grow warmer, loving orchids. But certainly the Masdevallia species are very popular, as are some of the others. They're absolutely amazing little things. And the world's expert on the pleurophthalid orchids, you may know, is a person named Carlisle Lure. You may know his name. He was a retired physician and was just obsessed with pleurophthalid orchids. And he wrote a whole series of monographs and books called Icones Pleurophthalidinarum. And in volume number 4, published by the Missouri Botanical Garden in 1987, Lure considered 3 genera that he thought were related to each other: Acostea, Condylago, and Porroglossum. So this is Porroglossum that we've been talking about. This is another species. It has the trigger on its lip, but so does Acostea. This is another pleurophthalid. Looks quite a bit different, but it also has that mobile or snap trap flower. You can see up above the lip in the open and the closed position. And then there's a third genus called Condylago. I happen to grow this one. It's not uncommon in cultivation. Really weird, fuzzy little flowers, but they also have the ability to snap shut. Their flower has a trapping mechanism. So Lure thought that all 3 of these were related, but when we started doing some DNA analysis and some genetic work, we came to realize that all 3 of these are completely unrelated to each other. There are at least 3 independent evolutionary origins for this complex floral snap trap. And you can see on the family tree to your right, I highlighted in red those 3 different groups. So they're unrelated to each other. They've evolved this mechanism separately. So I just think that's fascinating as an evolutionary biologist. Porroglossum, however, is related to things you would know, like Masdevallia, Dracula, Trisatella. They do share some overall floral symmetry in terms of their flower shapes. And so we do know where Porroglossum fits in the family tree of the orchid family. But for a small genus, there's only around 20, 25 species. Just look at the variation. I mean, every single one of these species of Porroglossum, to me, looks completely different from the others. There's very few hybrids that have been made. A couple have been made with Masdevallia and Dracula, but basically all of these are wild species that have taken on completely different forms in nature. So Lure in 1987 only knew about 19 species. There were only 19 described at that time, and he tried to divide them up into subgenera and sections. A little bit, and he presented a key for identifying those. A little bit later, a person named Aubryn in 2005 also tried to classify the different species of Porroglossum. And by that time, there were 35 known species. So just in a short amount of time, we've almost doubled the number of species known from this genus, and new ones continue to be described every year. One of my former PhD students is a guy named Alfonso Doucet. You might know his name. He publishes a lot of new species of Lepanthes and so forth, but he's described some new Porroglossums as well. So as we explore the Andes, we're finding more and more of them. I'd like to just profile some of the major groups of Porroglossum so you have a sense of their diversity. Probably the most beautiful are the 2 species within the subgenus Edwardia. So the one on the left with the big, beautiful red flower, that's Porroglossum edwardi. And then the one in the middle is very closely related, called Porroglossum lorraine. And what distinguishes these is that they have a very long creeping rhizome and then a very long inflorescence called a raceme, and the flower is at the very tip of that. These are probably the most difficult to cultivate. For some reason, they need to creep and crawl and have just the right substrate. But certainly they're the most beautiful. Another group, we call this the section echidnae. Do you know what an echidna is? That's a spiny animal in Australia. It's a marsupial. No, it's a monotreme. They're related to platypus. They're covered in spines, or something that's echinate means it's covered in spines. And so all of the species in this group have a pubescent or a spiny peduncle. The stalk that the flower sits on is covered in these hairs. So that is the section echidnae. And then there's another section that's called section porroglossum. What's special about this, or maybe not so special, is that the ovary of the flower twists, like most orchids, so that the lip is facing down, right? When we look at a typical orchid flower, it's because the ovary has twisted 180 degrees so that the lip is presented forward. And all of these, you can see that twisted ovary in the picture on the left, belong to that section. But then there's another group where the ovary, it doesn't just twist 180 degrees, it twists a full 360 degrees. So you can see the picture on the left there, that twisted ovary, it's contorted, right? So we call this section torti. It's a contorted flower, so that now the labellum, the lip, is pointing upward. It's kind of like a toilet bowl, to be honest, with a toilet seat pointing up. So the last group is called subsection brevisepala, and all of the species in this group have three veins running on the top of their upper sepal, and that dorsal upper sepal bends backward. It creates a hood and it bends backward naturally. So there's a whole bunch of species that have that sort of appearance. So we put all that together, and my student James and I sequenced DNA from all of the species of Porroglossum to see if that pattern held up in the evolutionary tree. And sort of, the subgenus Edwardi at the bottom seems to be the most primitive, and then we have other clusters of groups there. The problem with this particular study I'm showing you here is that it was very early work based on just one gene, one little fragment of DNA. And now we have the ability to do so much more than analyze just one gene at a time. So what James did was to add more genes. This particular figure is based on 5 different genetic markers, and he's getting better resolution of the tree. The branches are coming together, and they're having stronger statistical support, so that we have confidence in our result. So James had all of that information at hand when he started his PhD, and he had 5 different objectives. And I'm sorry about all the text here, but in bold, he wanted to quantify the kinematic variation, the movements. Number 2, he wanted to understand the genetic relationships. Number 3, he was looking for evolutionary shifts or patterns. And then finally, number 5, looking for what variables could be used to explain any pattern that we might find. And we were so fortunate to have a living laboratory available to us down in Ecuador. I hope some of you have had a chance to visit the nursery Equigenera. You've at least heard of them, I'm sure. Equigenera in Wallaceo, Ecuador is at 2,200 meters, and they have greenhouses full of porroglossums. And so what that allowed us to do was to take our video cameras down there, and basically a common garden experiment. Same elevation, same temperature, same humidity, all of the variables were identical. And what James did was to take 916 snap trap videos for 33 different species. In terms of the genetic analysis, I mentioned we can do more than one gene at a time now. down there in red circled was what we ended up analyzing 9,000 different genetic markers, representing over 32,899 mutations. A huge data set of genetic information that we could never have done, 10 years ago. So just real quickly to show this, I know it's probably not as interesting as the other part, but on the left was our original family tree based on 5 genes. It had short branches, low statistical support. It was more or less unresolved. On the right, sequencing the 32,000 genetic markers, we have a perfectly resolved tree, and every single branch receives 100% statistical support, except for the couple of them that are marked there with 97 and 90. So we feel really strong. We feel really confident that our understanding of the genetics now is very firm. And we go back to Luer's classification system and to Auburn's classification system, and we realize that they both kind of were right, but they both kind of were wrong. So the way our tree is broken up is into 3 major groups. The blue group, which is mostly that Brevisepala with the recurved hood. The red group, which is all the spiny ones, right? The echidna group. And then the Porroglossum group, the one with the twisted ovaries. The Snaptrap video data was really interesting. This is now looking on the bottom, the x-axis, and each of these rectangles is a different species. So we have 33 species. And then on the left, the scale is showing time in seconds. So how quickly do the flowers snap shut? So the ones down here in the bottom left corner, those are really fast. I mean, they're snapping shut in about 1 to 2 or 3 seconds for those flowers to close. The ones on the upper right corner are the slow snap traps. They're taking over 200 seconds. What is that? Like 2 minutes to be fully closed. And then we've got a couple of weird outliers in the pattern as well. So, we kind of scratched our head, like, why are there these different groups that some move fast and some move slow? Well, the genetics explains some of it. So everything in the blue group on the family tree, the Brevisepala group, those basically all tend to snap close very quickly. The only exception is that weird red-flowered Porroglossum edwardi. And what we think is happening there is it's moved away from fly pollination and probably moving more toward hummingbird or some other kind of insect. So it's a real outlier. It doesn't fit with its cousins. But the flower doesn't fit either. It's so different. So that isn't such a surprise. The echidna group, the 6 species listed here, all have those fuzzy flowers. They're a little bit slower, but they're also still pretty fast, between 1.5 seconds to 8, 8 seconds. And then we get to the big green group, the Porroglossum clade, as we call it. And all of these are slow movers, except for 2. These 2 little species shown here on the slide, Porroglossum miguelangeli and Porroglossum lycaenum, both of those are fast movers, but genetically related to the slow movers. So why? Like they should be, they're out of sync with their sisters and brothers. So we're trying to figure out if we could explain that. And I do have an explanation. I'll save it for just a minute. Remember, however, that in those 3 videos I showed you, it's not just about the start to finish time, it's also about the pattern of movement, the kinematics. And so we look at, for example, this kinematic plot, and we see that we touch the flower and we get a little trigger response at the beginning. And then we can see a very slow and steady movement through time before we get a pretty rapid snapping shut with kind of a curve at the end. So that's one kind of pattern. In the echidna clade, it's more or less the same visual. Maybe the amount of time is longer, but they still kind of follow the same general pattern. Whereas in the Porroglossum group, we see an initial trigger response at the beginning, then a slow period of not very little movement, then one little bump, right? A little bit of a movement. Maybe you don't want to scare the fly away, so you just move a little bit. Then a slow and steady closure before we get a really rapid response, just skyrockets straight up. And I guess the way to think about this, I was thinking about it as I drove over here this morning. It's kind of like the Olympics, right? So you've got speed skaters who some of them get out of the gate and they go fast the whole time and they lose the race. And others start out slow, but then they have a burst of speed at the end and they win the race. So it's not just about the start to finish time, it's the pattern of movement that you do during that interval. So we think we've documented all of this in great detail. Now, does it have any significance in terms of evolutionary biology? Well, using our genetic tree, and we color coded it as with these like hot and cold colors, blue being slow and red being fast. We think we can see a pattern here of some shifts, that in the evolutionary history of this group, there were some shifts from a fast flower, a fast ancestor, to a slow species, or from a slow ancestor to a fast descendant. So that's one way to visualize it. Here's another way to see that these shifts, these evolutionary shifts in the genetic tree. But I think this particular graph here really kind of sums it up the best. And James completely stumbled on this by accident. He decided to plot the elevation of each species on a graph. So along the bottom, the x-axis, you see that some species grow at around 1,200 meters, and others are growing way up high, over 2,200 meters in the Andes. And then on the y-axis, on the left there, we have the speed, the time to closure. So the 1 second, 2 seconds, 3 seconds, 4, 5 seconds. What we're seeing here is that the group of species that are slow movers, the ones that take their time to snap shut, they're all growing at low elevation. So those, I'm calling that the low and slow group. Whereas the ones that snap shut very quickly within 1 or 2 seconds, they're mostly growing at high elevation. So, I'm going to let you think about that for a minute. Like, why would you move? I don't know. When I'm cold, I move slowly. But these flowers, when they're cold, way up high, seem to be moving fast. So why might that be? We think that the explanation is that the higher you go in elevation, the fewer insects there are. There just aren't many pollinators way up high at 2,000 meters. So if a fly lands on you, you better snap shut quickly, because that might be your only chance to grab that pollinator. So we think it has to do somewhat with their biology. And a little bit of further evidence for that. Do you remember those two weird ones that I mentioned, Miguelangeli and Lycaenum? They should be up there in the green Porroglossum group. Their genetics say that they should be low and slow, but they're acting like the other group, and that's because they grow at high elevation. So they have moved into a high elevation habitat and adapted this fast movement strategy because of the environment and the need to capture pollinators. So we think that's kind of indirect evidence that our theory makes some sense. I hope I'm making sense there. Okay, so kind of summing all of this part up, I just want to say that I think this little genus, Porroglossum, that more people should be growing, is a great model system because it allowed us to consider orchid form and function together with pollinators, right? Their interaction with pollinators in their environment and thinking about elevation together with their genetics and their evolutionary history. And all of those things are interconnected with one another to explain this one little fascinating group of orchids. So I hope I've inspired you to maybe give it a try to grow some of these. One argument I make when I talk to orchid societies is that these are great for educational outreach. Children love Venus flytraps and carnivorous plants and bugs and anything that moves. Trying to get kids interested in orchids is sometimes a challenge, but if you can show them Porroglossum in action, they just love it. They eat it up. So, in fact, at the World Orchid Congress some years ago, one of the groups out of Europe had these educational display posters out in an educational exhibit about Porroglossum. And I thought it was really wonderful. They had beautiful graphics, and a lot of kids were looking at them. This was also a World Orchid Congress, the one back in Wallaceo, Ecuador, a long time ago. And Equigenera put out these glass display terrarium cabinets talking about Porroglossum, and the public was learning about them and seeing them. So those are just some ideas that your club might consider. And I will say that they're not difficult to grow, provided that you can give them the cool, humid conditions that replicate their high elevation habitats. And maybe down here in Kona, it's too warm, but you guys have elevation here on the big island of Hawaii. So I'm sure that those of you who live on the mountains would be able to grow these without too much trouble. Some of the most beautiful specimens I saw were at Colombo Orchideas near Medellin, Colombia. That's a nursery by Jose Posada. And my friend Elsie took me there, and they had thousands of Porroglossums just growing out under the shade house. So they just need humidity and cool temperatures. They also like shade. They're not full sun. They're almost growing in the dark, to be honest, like deep shade, cool, humid conditions. You can buy them through Equigenera. They're pretty cheap, $12, $20. And I happen to know that Tom Miranda placed an order for about 20 or 24 plants. We were hoping that we would be in person and that you would be inspired to buy them at the meeting today. So Tom has about 24 Porroglossums waiting for you to buy off of him. And I know if you make contact with Tom, he'd be happy to, set up a meeting and get you inspired to try growing Porroglossums. Back home in Madison, we have cool temperatures. We have frigid temperatures, so we basically have to grow them in special ways. My students Alfonso and Kelsey are famous for converting wine coolers into orchid terrariums. I don't know if you've seen this. So Kelsey is standing next to a wine cooler. You can set the temperature. You put an LED light in there and a computer fan and a timer, and you can grow all these high elevation pleurothalids right in your home, regardless of where you live. So my students can afford to buy wine coolers. I can't even do that. And so if you're interested in that, send me an email. That's my email address in the bottom corner, kmcameron@wisc.edu. And I'll send you this article that Alfonso wrote on how to modify a wine cooler for growing pleurothalids. Personally, I grow them in a reptile terrarium. It's a glass thing that has doors that open, and I have a little computer fan in there with lights on the outside. And it's in my basement, so it stays nice and cool. So mostly you just want to keep them humid and cool. And so with that, I will just acknowledge Equigenera Nursery, the National Science Foundation, and a number of different organizations that help to support our research. And again, to acknowledge James McDaniel, my PhD student who did most of this work. And I thank you for your attention. I'd be happy to answer any questions that you might have. You can type them in the chat or raise your hand or shout out loud. Thank you very much.
Thank you, Ken. I think if you unshare your screen, we can see everybody, and then we can unmute them. Larry, can we unmute the people that are raising their hands?
We should be able to, yes.
And feel free to type in the chat if you're shy. I know that a lot of people are.
Okay, so Ken, we need you to unshare your screen.
Oh, I did unshare it. Are you seeing
Oh,
anything?
no, okay, it's okay. I'm seeing you as the speaker. That's fine.
Okay.
Let me-
Okay.
Yeah, if you go to I'm on gallery, so
Yep.
I can see everybody. Does anyone have any questions for Ken?
Does anyone grow Porroglossum? Do you have pleurophthalid people in your club?
So I was just down in my garden looking at some really strange orchids.
Yeah.
Because I just buy when I think, oh, this one's really cool, I buy it and then I forget what it is.
Yeah.
Although I do have the name tags on it. And I think I saw two on your screen. So I wrote down the names and I'm going to go down to the garden and see because
Good.
I live at 1,700 feet.
Yeah.
And it's cool. The Mastovalias love it up there.
You
so
need
I've got to see if I have
Porroglossums.
some.
You need those. They're really amazing little flowers.
Oh, there's a new chat. Very interesting. Thank you.
You're welcome.
And Ken, we do have some members who live up in Volcano at 4,000 feet or so elevation that and they can definitely grow cool growing orchids, but I don't know if any of them are here for
They may have lost their internet. We're on that side.
It's great that Larry recorded this so that our members who couldn't log on now can see it. And then again, as Ken said, Tom bought some of these Porroglossums from Equigeneri, and he does have them. They arrive this weekend. So we'll have to connect with Tom. However, Tom's going to be leaving to go to a World Orchid Conference, so he won't be around. Anyway, we'll get these. Someday we'll be able to touch these orchids that Tom ordered.
You know, one thing I failed to mention is that they also have beautiful leaves. I showed kind of a pattern, but if you grow them well, they take on a blue iridescent color. I don't know if you've ever seen certain ferns and begonias that can take on a blue iridescent color. If you grow them in low light and they're happy, a lot of Porroglossums have a bluish colored leaf. So even without flowers, they're just beautiful to have around.
All right. Okay, I don't see any questions. We want to thank you again, Ken,
You're
for
welcome.
weathering the storm.
No, thank you. And again, I'm really sorry that I didn't get to meet you all. Next time.
Oh, wait, there's some more. Okay. Matthew asked, what are the warmer, lower elevation species?
You know, they're all still high elevation. It's not like Masdevalias where you've got some that are high and some that are low. I would pretty much say that all Porroglossums need the cooler temperatures. So it's a kind of a fine line, but I see another question here, whether you can bring them down to Hilo for an exhibit, and that would be fine. You know, they can take, you know, a week or so of warmer temperature as long as they're staying hydrated and moist. So I wouldn't worry about bringing them to a show for a few days at a time. And like I said, whenever I've seen them in exhibits as used in an educational capacity, kids really like them. They're just really fun fun to play with. Someone else, Teresa, is asking if they can survive without their insects. So remember that the insect in this case is not for nourishment like a Venus flytrap or a carnivorous plant. It's there to collect the pollen and hopefully fly off to another flower and get trapped and deposit the pollen. So the insects are there to pollinate the flowers, and it probably is a very rare chance, even though this is a remarkable strategy that's evolved. I'm just going to make this up. 1 in 1,000 actually works, that it actually catches the insect at the right moment. It's one reason in nature we just don't find seed capsules very often on most orchids. The strategy is very complex. It fails most of the time, but when it works, that one time out of 1,000, you get millions of seeds. So it's a strategy that has cost benefit aspects to it, but obviously it works very well because the orchids are the largest family of plants on Earth. Jan is asking how long the traps stay shut. That's a really great question. And after we finished this research, we had more questions than we answered. That was one of them. We realized that if we were ever to do this study, a follow-up, we would time how long it takes the flower to reopen. Very often it takes a full day. Sometimes we find that all of the flowers are closed at night for some reason. Like the plant closes up and then they reset again in the morning. And we don't know if that pattern holds up for all of the species. The other thing that we wondered is, can they wear out, right? Like if I jog a mile, I'm kind of tired and I'm not sure I want to run another mile. Or as you get older, is the flower ages, does it slow down or does it lose its trapping ability? So those are all really good questions, and we don't have answers to them. But oftentimes the flower stays shut for several hours, if not overnight.
So
And you're
Andrea
asking if
has,
they should be in a glass case, and I would say yes. Unless you live at a high elevation where it stays cool, then you probably don't need a glass case, but a lot of people probably would. I'm sorry, somebody was about to ask something.
Andrea had raised her hand. Andrea?
Yeah. So can you hear me?
Yes.
Yep.
Okay. Um, so I'm at 1,300 feet elevation, and we're consistently about 5 degrees cooler than sea level. It does get warm in the summertime, though, and I'm wondering, would these be best grown potted outdoors, strapped to a tree outdoors, or
Like, how warm does it get for you?
75 maybe, 77? I don't know.
But usually lower, less than that?
And then, yeah, it's always, I mean, it's 50 degrees at night sometimes up here, but I
I
have,
would imagine you can grow them outside, to be honest, as long as they're staying humid.
okay,
Like anything in the 50s at night to the low 70s in the day would be perfect.
okay.
And on those couple of rare weeks where you get a little bit warmer, just make sure they're misted and staying, moist, that
And
they
then
don't dry out.
indoor kind of pots like this, would that be okay?
I always grow mine in mesh pots,
Mesh.
so that and I grow them in moss,
These are
in sphagnum moss.
these are separate pot inside, so I could do maybe a mesh pot inside the hanging. Is that, or is there something wrong with that? That wouldn't work.
That little ball that you've got there of kind of twigs,
Yeah,
is that glass or just open?
this is just wire mesh, like
See,
a
I think I would put a porroglossum inside of that,
okay,
and
cool.
I would mist it every day.
Great. Okay, thanks for the ideas. I
Yeah.
appreciate this. This was really awesome. This was like I love this. Thank you so much for
Oh,
joining us.
you're welcome. Thank you.
I think Jan had another question. How long, or somebody, L, had a question. How long do the flowers usually last?
Oh, they last a long time. I'm gonna say you get 2 weeks, maybe around 2 weeks, but then the same inflorescence will make another bud. Like it'll be successive flowering on the same stalk. You get maybe two or three. So yeah, I don't a month. Like it might stay in flower for a month once it gets started. They last quite a while.
All right. Are there any more questions for Ken? Now, what is that, Jeff?
This is Macropodanthus crutzii. So anyway, this is Trichopilia suavis. And it smells nice.
Wow, that's beautiful.
And such phylogeny blasting in the wind.
Cool.
Thank you for your talk. It's very interesting.
Yes, Ken, your talk was very interesting.
Oh, good. Thanks.
Does anyone have anything else to say?
I had one more question. Will we have posters of the orchid show ahead of time?
What did you say? The
Should-
posters for the orchid show? Yeah, we'll be doing that.
For the,
Now,
for the
Andrea,
artwork. Yeah.
you'd love to come to our show committee meeting on March 24th, Tuesday,
Oh, it's this soon. Okay, okay.
and find out all about the show. I don't think I've ever met you before.
I've been at a few. I just joined last show.
Okay.
So March 24th at the Senior Center.
Yeah, it's a Tuesday at 4:30.
Tuesday at 4:30.
I'm trying to unmute. I mean, mute.
All right, okay, I'm going to close the meeting. Oh, I'm going to close the meeting if everyone's finished then.